When did the universe start? What makes up the universe? When did life start? Is there life outside our planet? And why not? And what is our purpose in life? Those questions are being answered by the branches of science: physics, chemistry, astronomy, social science, and of course, biology. But when it comes to life and how it originated, biologists investigate our origins by examining our biological processes, our cells, our smallest components, and even the chemistry behind them. In order to understand better our origins, biologists have performed experiments simulating the formation of living organisms, cells, and their components. It's like finding out the recipe by reverse engineering life itself and trying out what works. This episode in Earth and Life Science Rewind, we will discuss the experiments on the origin of life. Greetings, senior high school students. I am teacher Gian, and welcome to Earth and Life Science Rewind, video reviewers for Earth and Life Science. We will study how our understanding of the origin of life evolved from the experiments of biologists regarding the origin of life. Our objective is to describe classic experiments that model conditions which may have enabled the first life forms to evolve. The earliest theory on the origin of life started with Aristotle, believing that living things came from non-living matter, which is termed a biogenesis or spontaneous generation. People during his time believed that mud, water, and dead leaves have given life to animals like frogs and fishes. Rotten meat gives life to flies and other insects. And ants literally came from rotten food and expired bread. But it wasn't until 1668 when Francisco Redi proved this to be untrue by his meat and mesh wire experiment. He disproved the theory that rotten meat gave life to flies. How? He created 3 specimens of rotten meat. One that it has an open container, one that has a tight seal with no air that can enter or exit, and a gaze-covered rotten meat in which air can enter. The result is that only the open jar had flies around the rotten meat, which means that it was the then traveling flies from other places that smelled the meat and brought eggs to it. So no life came out of the rotten meat itself. Another development was the discovery of microorganisms by Anthony van Leeuwenhoek. In his self-made microscope, he noticed various living organisms which are microscopic, very small, in water samples, such as mosquito larvae, protozoans, sperm cell paramecia, and it led to the idea that all living matter may have started from bacteria, fungi, protozoa, and other microorganisms. Almost a century later, a biologist named John Needham discovered that microorganisms flourished on soup that has been heated or exposed in air. Needham heated the broth or the soup and let it expose in the air for several days. After then, he observed microorganisms in the fluid, contradicting Francisco Redi and establishing again the concept of spontaneous generation. He thought that lifeless broth or soup gave life to the microbes. Decades after, Lazzaro Spallanzani performed hundreds of carefully executed experiments using heated broth, simulating Needham's experiment. Except for sealing the jar, broth in sealed jars developed no microorganisms. So the growth of microorganisms was only because it was exposed to the air. The broth itself did not produce life. He concluded that the microorganisms were present in the air already and settled on the broth. So he disproved the proposal of John Needham regarding spontaneous generation. So with Spallanzani's conclusions, heat, air, and moisture are key factors for life. Microorganisms did not appear on sealed containers. And with that, another concept, hypothesis, started, the biogenesis hypothesis, that living matter arise only from other living matter. And with the concept of biogenesis, microorganisms being present in the air, that is why food products are needed to be tightly sealed, sterilized, and refrigerated to control microbial growth. Almost a century later, Louis Pasteur cemented the concept of biogenesis and proved by demonstrating bacterial growth in an S-flask experiment. He tried to copy Spallanzani's experiment except for being sealed. He put an S-shaped tube in which air can enter, but dust and particles can settle on the middle of the S tube. On the first specimen, he heated the broth, let the flask sit for days, And although air can go in and out of the flask because of S-shaped tube, there was no bacterial growth. But when he heated the broth and removed the neck, after several days, there was bacterial growth. So it means because of the S-tube that traps dust and bacteria, but allowing air to pass through, it means that bacteria grew not from the air, but from earlier bacteria. So what does it mean? People believed before this that microbes and life forms grow in the air, out of nowhere. But with this experiment, Pasteur proved that bacteria did not grow in the air inside the ice flask. It means bacteria grow from other bacteria through reproduction. If the environment has no bacteria can grow from it. So it is possible to create an environment which it is bacteria-free or sterilized. In fact, it was Louis Pasteur that created that concept of pasteurization and sterilization that helped increase the quality and sanitation of our food products. So pretty much, the biogenesis concept in which life came from other life is already established, and the spontaneous generation theory is already debunked. Is it for good? Let us find out. Biochemists Operant and Haldane gave the idea in 1924 that microorganisms evolved from the mixing of chemicals during the early stages of Earth. He called this theory the Primordial Soup theory. So it was kind of groundbreaking. Imagine that spontaneous generation theory has been disproved, but a group of scientists tried to propose it again, but in another twist. It was chemicals, to be specific. In 1953, chemists Miller and Urey performed their famous experiment. They mixed water, ammonia, methane, hydrogen gas, and electric sparks to produce organic materials that are necessary for life. This is to support the claim of Operant earlier that it were the chemicals that formed life. First part of the experiment was water was boiled and added with chemicals which are inorganic, carbon dioxide, ammonia, and methane. Again, inorganic. And then they heated the mixture and put it in a chamber wherein they simulated lightning. So they added electricity there. After electrocuting the solution, they condensed it into liquid form, and the dark mixture contained amino acids and biomolecules that are needed for life. Organic compounds. This was groundbreaking in the field of biology because for the first time ever, organic compounds are created within a laboratory using inorganic compounds. So the organic compounds that are present in life forms, organisms, are created artificially. Almost a decade later, Sidney Fox, which is inspired by the findings of Miller-Urey, heated a mixture of amino acids and produced proteinoids. What are proteinoids? They look like cells. They are lifeless molecules and spherical in shape, and they exhibit properties which look like cells. This proposes again that biomolecules came from inorganic substances, and when you mix organic substances, they can form into sphere shape that can resemble cells. The only missing factor here in the field of biology is that how did it come to light? These discoveries have become the basis for most developments on biological medicine, laboratory meat, and lab-produced hormones. Manufacture of insulin is inspired by these experiments. The creation of artificial meat, in which we don't need to kill animals for meat any longer, is being developed by creation of nutrients and organic substances from sample cells. Hormones like human growth hormones and melatonin are being manufactured artificially. So to summarize our video reviewer for today, it started with Aristotle and his concept of a biogenesis or spontaneous generation, wherein life came from no life. Francisco already tried to debunk the biogenesis theory by his meat and mesh wire experiment. Antony van Leeuwenhoek discovered microorganisms in water using his self-made microscope and led the idea that all living matter started from bacteria, fungi, and protozoa. John Needham discovered that microorganisms flourished on soup that has been heated or exposed in the air. Later debunked by Spallanzani that concluded that microorganisms were present in the air already. Louis Pasteur cemented the biogenesis concept, life came from no life, and proved by demonstrating no bacterial growth in an S-flask. Operant and Haldane gave the idea of the primordial soup to be supported by Miller and Urey in 1953 with their famous experiment. Sidney Fox, inspired by the findings of Miller-Urey, heated a mixture of amino acids and produced sphere-shaped proteinoids, which look like cells. Let us test your knowledge in this video reviewer. Please pause this video and try to answer this 5-item quiz. You can resume the video to find the answers and check yourself. Video resumes in 5, 4, 3, 2, 1. Here are the answers in our Test Your Knowledge portion. Thank you for joining us again in another episode of Earth and Life Science Rewind on the theories on the origin of life. This is Teacher Gian, and I hope to see you next time and study smart.